Afternoon, everybody. I'm Steve DeMello, Director of Healthcare here at Citrus. Welcome to the Research Exchange. Also, welcome to our online viewers. Couple of announcements. The I for Energy talk this week, Friday at noon, is by John Elliott, the Chief Sustainability Officer at Lawrence Berkeley National Lab. CLDA is April 20th, and the campus will be open with numerous talks and exhibits. You can check it out online at calday.berkeley.edu. And last reminder, the Big Ideas at Berkeley Grand Prize Pitch Day will take place on April 25th from 2 to 5 p.m. in B100 of Bloom Hall. This is an inspiring event that brings together some of the best big ideas from the 2013 contest. Select group of finalist teams will be given the opportunity to present their ideas publicly for a chance to win additional funding and development support. Brain-machine interfaces hold great potential to aid large numbers of people with neurological disorders. Much of the pioneering work in this field is happening at UC Berkeley and UCSF, and we're really fortunate to have one of the leaders of that effort here with us today. Jose Carmena is Associate Professor of Electrical Engineering and Neuroscience at UC Berkeley and co-director of the Center for Neural Engineering and Prostheses at UC Berkeley and UCSF. His research program in neural engineering and systems neuroscience is aimed at understanding the neural basis of sensomotor learning and control and at building the science and engineering base that will allow for future development. Please join me in welcoming Professor Jose Carmena.
Hello, everyone. Can you hear me? Great. Thank you for coming. It's amazing what a free lunch does in terms of crowd. So in 40 minutes or so, I think I have 40 minutes. I'm going to try to summarize what's going on in the lab right now in the exciting field of brain-machine interfaces. And to start with, I'll thank the wonderful group of people I have the luck to work with. In case I, normally rush this slide at the very end, and I think it's very important to highlight all the tremendous effort that these folks are really the ones at the bench doing all the things that you will see, and in some cases you will not see because of the lack of time. And in yellow out the folks that are already graduating from the lab. Okay, so to pitch the idea that most of you are familiar with in this brain-machine interfaces, or BMI from now on, field, the idea is to restore motor function or sensory motor function in people with all sorts of disabilities. Just looking at numbers in spinal cord injury, there are about more than 1 million patients or impacted people like family relatives and so on in the U.S. alone. So one solution came about a decade ago in terms of a promising pathway to restore this function, a little bit more than a decade, I will say, although it has been in the mind of many people from several decades, is this field in which, irrespective of which kind of signal we record from the brain, meaning in an invasive or non-invasive way, and today we'll be talking all about invasive technology, and what kind of information we transform or interpret or decode from these signals. Presumably we should be able to generate motor commands to control all sorts of devices, like computer cursors in the screen, or steer a wheelchair, or more challenging, dexterous movement of a prosthetic limb or exoskeleton to walk, and so on. So that's the main idea. Just to give you, in one slide, 2 examples of very successful neuroprosthetic devices. One is the flagship of the field, which is the cochlear implant, which is basically now implanted in more than 150,000 users worldwide, including infants, okay, and allows you to, basically this allows to restore hearing. The other one is more recent, still widely used right now worldwide as well, and is the deep brain stimulator, mostly used for Parkinson's disease, but these days is also being investigated for other disorders more related to conduct and behavior. So both are invasive. This is a sensory device. It gets all the way into the basilar membrane in your inner ear. The other one is for motor purposes, but basically you can see that is actually as invasive as it can be, right? You insert this electrode all the way into the subthalamic nucleus, and then you stimulate electrical current, and that actually alleviates the tremor in patients in which drugs don't do the job anymore. Okay. zooming in a little bit in the problem that we are interested as a lab, both from neuroprosthetic applications as well as from basic neuroscience, is the problem of the neural control of movement, or how the brain controls movement. And here you have a simplified diagram with all the basic elements from, the main building blocks, going from the brain and brainstem, spinal cord, and the biomechanical plant, all the way to some kind of behavior that's being performed in the real world. And in particular, in the lab, we're centered around the question of how the brain can learn to perform novel actions irrespective of physical movement. Okay, for different reasons, but as you can imagine, the irrespective of natural physical movement is related to the fact that at some point, people might not be able to move because of, let's say, a lesion in the spinal cord injury, and these sensory and motor pathways are interrupted. So then can the brain learn to perform new actions, meaning actions that have never been learned to perform before, and this includes with an effector that typically might be disembodied, like not part of your body. In some cases, it will still be a remaining part of your body, like for amputees, they will still have part of the limb, or in case of a stroke, they might still have residual limb movement, but in many cases, it will be, like for example, in a spinal cord injury, it will be controlling a robotic arm to feed themselves or something like that, and that's what we refer to as disembodied control. And within all this BMI spectrum, we're very, very interested, or perhaps the largest concentration of work in the lab hones around the question of how the brain can learn to adapt to the machine, right? This long-term long-term plasticity, long-term learning of how to actually drive these actuators, these disembodied actuators. And there has been a lot of work being done in the last decade. Here I'm only citing major papers in invasive BMI in rodent, human, and non-human primates, but there are more and more coming pretty much every week. So I will tell you a little bit about the methods that we use in the lab in one slide. So we're mostly an experimental lab. We work with non-human primates and rodents as animal subjects. These animals get implanted at some point during the experimental design or behavioral training phase, with these arrays of microwires that you see there. These are basically tungsten microwires of 35 micron in diameter, and they go implanted about 2 millimeters into the motor sensory cortex of the monkey or rat. And here you see two areas that typically we implant in the macaque brain, in the primary motor cortex and dorsal premotor cortex, which are very relevant to movement or for action. And just to give you a glimpse of what kind of data we work with, from each of these microwires that are chronically implanted in the brain, we can record the activity of the extracellular discharge of single neurons. And you see here 3 cases of electrodes recording 2 neurons, 4 neurons, and 1 huge neuron as well. And what you are seeing here is superimposed the action potentials or the discharge of these neurons. So the neuron can be silent, and then when it fires, that's what we call a spike or an action potential, and that's what you are seeing there. Okay. So the beauty of this technique is that you have some sort of spatiotemporal resolution, so you get to see like a little window into the brain, like what part of the circuit, a tiny part of the circuit, is doing while the animal is the awake behaving animal is doing some kind of a behavioral task. And this lasts for several months, even years. So basically, it's very good for neuroscience. Is good enough for proof of concept in neuroprosthetics. It's not good, not even close, for clinically viable prosthetics. Okay, so that's one of the bottlenecks we'll talk in a minute. Just as I mentioned in one slide, the glimpse of what goes on in the lab with this. Basically, these methods of this technology, we have different experimental paradigms with these animals. Either they're doing a natural motor task, for example, or a BMI task, which is what we will see in the rest of the talk. And we can, with this, look at different questions of motor and sensory encoding, a lot of decoding and adaptation in the brain, and also more recently very exciting questions about how the oscillatory activity in the brain, these brain rhythms, relate to the actual activity of individual neurons in the brain in different areas. All right, so let me say a little bit about what the state of the art is actually in humans, not from our lab, okay, from other labs, and what are the challenges. And I have dichotomized this in two two main challenges. What I refer to as challenge 1, which is what goes inside the brain, so the neural implant, the implantable device, so to speak, like the DBS or the cochlear implant that we saw before. So these are typical. This is exactly the array that today is approved for human use. This is called the Utah array, or now BlackRock array, which is the company that commercializes it. And this is a MEMS-based device with silicon spikes. It's about 96 spikes there in a 4x4 millimeter footprint. And so as you can see, it's pretty bulky. It's tethered. Okay, there's no wireless solution yet, at least that is FDA approved. And this is how it looks how the end product looks in 3 different patients here. So this is the connector, basically, that is actually plugged into the little pedestal that you see there, which connects to what is inside the brain or under the skull, which is the array. Okay, so the other problem that I will be talking about is what I refer to as challenge 2, which is what can you do with the activity that you record from these arrays? Okay. So again, inside the brain, now outside the brain. What kind of control can you achieve, right? And I think it's fair to say that there's a limited today and a natural control being achieved by a few clinical trials going on mostly in Boston and Pittsburgh. And this is the most recent one from Andy Schwartz's group and others. Showing, this is like the bottle half empty, half full, right? So it's extremely exciting for the field to see this already happening in humans. At the same time, it's not good enough. And one wonders whether it's actually even the risk-benefit ratio for the patient, whether it's actually good or not, right? So we need to think about how to drive the performance achieved by. You actually can see these videos are easily accessible on YouTube. For the sake of time, I'm just gonna skip through them, but basically you can see that it's good enough to prove the concept that a paralyzed person or a stroke patient actually can do the simple task like reaching for a bottle of water, but is far away from what we call as natural or naturalistic control or skillful control. Okay, so what is the opportunity? As I mentioned, inside the brain of challenge 1, pretty much every major university in the U.S. is working in coming up with their own neural interface system. Berkeley, of course, is doing that very aggressively. This is mainly the work of Michel Maharvis, Jan Rabai, and Elad Alon, among others. And what we call this neural dust, which is basically a radical new way of sample large numbers of cells in the brain. So everybody is trying to increase dramatically the density of these recordings, while at the same time dealing with issues about safety and so on, meaning making everything wireless, ultra-low power. So basically you put it in the brain and you can leave it there for decades, like the pacemaker, for example. So what will be the pacemaker for the brain? Okay. With respect to challenge 2, which is what the rest of the talk is going to be about, the opportunity or what remains to be seen or areas in which students, postdocs, different groups around the world working on very aggressively is, as I mentioned, okay, we get different kind of signals. In this case, today we'll be talking all about spikes, okay, the discharge of these neurons. So what kind of algorithms we should use, where should the learning take place in the brain, in the decoder, and so on. What kind of feedback is the patient going to get? And with this, I mean, the rest of the talk you will see work based on visual feedback, which is the dominant modality that has been done so far, among other things, because you can take it already for free. So if the patient can see, then you have a way of seeing the state of the prosthetic control at every time stamp and be able to adapt accordingly. Okay, so the brain will be able to modify or correct for errors based on this visual feedback. But obviously, that's presumably not enough, or the field is moving towards upgrading the system with what we refer to as somatosensory and feedback. So tactile, proprioception, what the robotic arm is grasping, and so on. And for that, there are different approaches, but one of which is to stimulate electrically or optically, maybe at some point, in neurons in the brain and get the brain to learn about that. So in the rest of the talk, I will be talking about what I'm calling like a wish list of what we would like to see in this neuroprosthetic design in terms of how to build what the future neuroprosthetic system should have, which is what we're working on. So we're working more on in the lab in the neural basis or the neurophysiology of action, and basically try to come up with design rules or principles that will be very important to translate them into clinical practice. So 3 points. We would like to have the equivalent to what we call a motor memory, right? Like you learn to drive your car, to play the piano, and then after you're proficient at it, you sit in the piano and you play, or you jump in your car. And not only you can drive without thinking, you can even multitask and talk on your phone or whatever, right? So presumably there is some sort of motor memory or after the acquisition of that skill that is being recalled readily every time you need it, right? So the question is, can we think of something similar in the context of a disembodied device? Let's say a computer cursor that you want to control in the screen by thinking without moving any part of your body, among other things, because maybe you can move. Or any kind of actuator. Okay, but again, we're talking about a disembodied. Something is not part of your body. Second important point, we would like to account on built-in machinery, so existing circuits in your brain that are being used for natural motor movements, meaning to move, let's say, your natural or native arm. But in this case, we like to take advantage of those circuits that are there, so you don't have to relearn the whole thing, because maybe for 2 dimensions it's okay, but to control 24 degrees of freedom from shoulders to fingertips to do some kind of movement, it took millions of years of evolution for the first mammal to learn how to perform a reach. They used to reach with the mouth, right? So the biomechanical plan is extremely complicated. So we might not want to reproduce that about a prosthetic actuator. Hopefully we would like to exploit what is in the brain, even though it's not to learn how to control part of your own brain, of your body. And the third point is like, so all these two gravitate around what can we account on the brain. Okay, what can the brain do in this specific problem? Third point is about, okay, but we also have machine learning and a lot of signal processing techniques that we could apply to accelerate learning, boost learning, and how these two guys should talk to each other. This co-adaptive system, right? Like two learners in the same closed loop system. So what has been done about that, and what will be the best way, or a good way at least, to pursue pushing both? Okay, and one comment about the plan, because as you can see here also, before the feedback, there will be some kind of actuator. And by no means, I'm not trying to imply that the plan is not important. It's extremely important, but in a way, being critic with our own field, I think it's fair to say that what is available today in terms of both commercially or in research labs, in terms of plans, in this case for prosthetic arms, basically exceeds the ability of BMIs to control them, meaning what can we do with this prosthetic implanted device in terms of controlling these limbs. Okay, there are too many degree of freedom already to control. Again, this is an extremely important field, and also in the exoskeleton device realm, there is not much going on, and so we actually need a big push there. And in fact, there is a big NSF-funded effort on campus with Claire Tomlin and Tommy Suka in mechanical engineering to control multi-degree of freedom exoskeleton that we are actually involved with. And but anyway, the point for this talk is that we are going to be focusing on the brain and not in the robot. Okay, as quoted by a colleague at MIT, Neville Hogan. Okay, there's a lot to be learned yet about how we can control these devices. Okay, so I will start with what I will refer to as neuroprosthetic skill, which is a definition I've made myself just to clarify what do we mean with neuroprosthetic, which I always put in front of skill, because otherwise motor skill by default it will be understood as natural motor learning and skillful movement of your own natural limb and so on. So here we're talking about proficient, readily recall control of a disembodied actuator, something that's not part of your own body, irrespective of natural physical movement. This means that there could be physical movement, but the point is that this is not necessary, or it should not be a requirement for the control. Okay, so it's not that everybody has to be immobile. Maybe a monkey or a human patient controls the device better by performing some kind of muscle twitch. That's fine, but the point is that the brain has to learn how to control it by volitional control of neural activity, at least part of it. So you can think of this like creating functional circuits for action. And with circuits, I mean this circuit for control, right? Which this cartoon represents the neural activity being recorded from the brain, the transform algorithm, which basically is this. I will refer also about this decoder as the spinal cord for prosthetic function, because in a way, you can see in this loop, it represents the spinal cord, right? So if you think like this is the neural activity in the brain, the plant is somewhere here. In this case, it's a computer cursor, right? This is a virtual plant, but you can think of a robotic arm here. In between the two, we have a spinal cord. So this is a many-to-few transform, simpler than the spinal cord, but subserves the same role. And a beauty of this paradigm, especially for neuroscience, I have to say, is that you actually define, the experimenter defines the output of the brain. In the natural case, in that cartoon that you saw before, there are many structures that project downstream to the spinal cord, all the way from cortical area, subcortical, brainstem, and so on, and lots of loops. And it's an extremely complicated hierarchy of areas. In here, and obviously we cannot observe all of them. In the BMI case, you get to select which neurons are going to output the brain. So by definition, that's the output for action. Okay, and then obviously you're going to observe some other cells or in different areas. So like part of the circuit, if you like. Okay, so let's go back in time about 11 years or so. This is in the days of where the field was trying to prove the concept that you can actually close the loop. You can have prosthetic control or volitional control of a computer cursor through neural activity, okay, in the absence of physical movement. So this is work, in this case, this is work I did with Miguel Nicolelis, when I was a postdoc at Duke in his lab. And what you are seeing here is a macaque monkey performing, oops, sorry, I shouldn't touch this while I play a video because it happens all the time. Let me, yeah. So what you see here is a macaque monkey performing what we call a reaching task, which is basically moving this manipulandum that controls that little cursor to hit a target, and it gets a user word every time it hits a target. So this is a manual task. And during the performance of the task, the animal is being recorded in different areas of the brain, motor sensory areas, and the activity that we are recording is linearly regressed to parameters of movement, in this case, position and velocity of the hand. Okay, and the testing part of the experiment that other groups were also working on those years as well is, can the animal, in the absence of physical movement, in this case, as you can see here, there's no joystick, the arm is resting there, can the animal perform the task now by thinking or looking at the screen? So basically what we call BMI mode. You can see that the animal can do it. The performance is not as good as manually, but very importantly, there is no arm movement, as you can see here. Okay, so these are EMGs of 3 muscle groups during the brain control phase. This is during manual control. You can see that the animal is actively moving the arm. It's actually resting the arm there. It's not trying to find contractions of muscles that will actually drive movement of the cursor. So, moreover, we could observe that there is actually an increase through training of through what we refer to as biofeedback on how the animal is doing. So this is in the moment that we switch to brain control mode or BMI mode, where during the course of an hour, you can see that the percentage of trials correct increases. Okay? So the brain is getting better and better through training, which is very exciting. Problem with that is that as you increase the complexity of the task, this is the first one that you just saw, so the video you just saw, and in red you have the BMI control. The animal does pretty decent, a little bit worse than the manual control, but pretty good. But the moment you do, and I will skip videos for the sake of time, a harder task like reach and grasp, a virtual object, okay, the performance in brain control drops significantly. And it will be very easy, basically, to make this flatten out and the animal not being able to do it at all. Because, so the question is why, right? So doing the experiment, you realize how sensitive performance is if you make the task a little bit harder. So yeah, on one side, again, the bottle half full, half empty. On one side, you prove the concept. The animal can do it. On the other hand, you worry about how do you scale up in complexity. What's the way to go? So if we look at what happens in the natural motor movement literature, there's a lot of evidence showing that obviously practice makes you better, but also that that is this kind of what I mentioned before, this concept of motor memory stabilization and after learning, right? So there's some kind of a map in the brain or a memory that facilitates this performance. The question here is whether in the context of a BMI setting with no arm movement, as we see again our cartoon here, can we actually expect the same? So our working hypothesis being whether if we keep this circuit stable, that will facilitate retention of what is being learned from day to day. What do we mean with stable? What we mean is if every day we could keep the same neural activity, the same channels, and we will use the same decoder, okay, while the animal is learning to perform the task, hopefully we'll expect that the animal will be able to start from where he left it the previous day and so on. In other words, get better to training and eventually becoming proficient at the task and being able to recall readily at the beginning of the task performance that level of achievement. Problem with this is that this is trivial. You just need to keep your same decoder. This is not trivial at all. This depends on having at least on those days, it requires having a very good recording quality for many days, right? The previous work, I should say, including ours, which basically every day it will retrain whichever activity was available in our implants to generate a new decoder to perform this task, right? And I will not get into details about why that was maybe thought on those days to be the way to do it, but it was definitely a limitation of the technique as well. You just can't decide when you can choose stable activity here. We were blessed in our first animal subjects in Berkeley, and we had very, very good quality in the recordings, so we went for it. And basically here you can see 15 channels with the same neural activity in day 1, day 10, and day 20. Okay, so this is what we refer to as stable recordings. Okay, and then basically what we did is we generated one decoder the first day, which is summarized here by this basically linear neural network, and then kept that for the rest of the experiment. And just to give you a summary of what we are doing here, just to make sure that everybody follows, this cartoon represents the network in the brain, okay, let's say motor cortex. We implant an array, which is this black box. In blue you have some cells that we can record from, and an arbitrary number of these cells, the red guys there, haven't really changed their properties from the last 2 days. So we decided to select them and expect that they're going to be stable for 1 or 2 more weeks, okay, which is not in our control, obviously. I'll skip that for the sake of time and then show you what happens in the first day that we put this guy to do brain control. Okay, and this is a very painful video because the animal knows how to perform this task really well manually. It's like a machine doing thousands of trials every day. And the task is basically manually, it consists of driving the cursor from the center target with the arm to a peripheral target. That's what we call a trial. It will get a user reward for that, and it will start again, initiating a trial, going to the center. So the animal knows the rules of the task, but it has never done it without physical movement, just basically looking at the screen. So this is the very first day. And what you see is the cursor being controlled by neural activity, trying to get to the center to initiate a trial, which he achieved there, but now he cannot reach to the peripheral target. Okay, again, he's trying, but he just doesn't know how to do it. This is jumping to another target, same thing. He managed to initiate a trial, but he cannot reach there. He's trying to figure out how do I control this cursor. And then if you compare that performance with this, let's say day 11, you can see the animal going and being able to hit every target. When he gets here, it's just enjoying the user reward, like disconnecting from the task for a couple of seconds, going back to the center to initiate a trial. If he misses, he can correct and go back and hit, and so on. So they can do this now for hours. Okay, so what is dramatic here is to compare the pre and post, right? So there are no biomechanics involved. The animal is not instructed to go in a straight line or anything like that. It's just hit that target, you get your user reward. That's the solution that the brain found for that. Okay, that's good enough for us if we can see any evidence of this skill acquisition, which we see not only in the learning curves of the two animals. You can see how it gets better and then it plateaus at 100%, and also becoming faster at hitting the targets, but also if we zoom in on what happens in a given day, right? So in day 2, when it doesn't know how to do the task yet, this shows you the performance, which is really bad. At some point, it starts to kick in, and the animal gets engaged. In day 4, it's already at about 45% accurate, but from the beginning, it finishes around 60. After day 6, you can see starting from the get-go at 100% all the way till the end. So it's recalling what it was learned before. if we look at what happens in the brain, so we have 15 neurons in this case that I showed you before. That's the output of this massive neural network because the whole brain is available there. But the output is this, is made by these 15 neurons. And the way to interpret this color map, basically, this is showing what we call the tuning or preferred direction of firing for a given neuron. So in red means that the neuron fires a lot for a given direction of movement. In this case of the cursor, in the natural case, will be of the hand of the arm, for example. And this is when the animal doesn't know how to do the task. If you compare how dissimilar is with the solution after that the brain adopts after learning, and how similar or identical is thereafter, right? So you can see before learning and after learning, that's what we call the motor memory, or the engram, if you like, the signature of this is what these cells have to do every time the animal is put in the chair to perform the task. Obviously, these cells do something different, and they fire in different ways when the animal is back in the cage. But when it's performing the task, this is what it has to recall in order to be able to perform the task. And you can see that red means similarity in all these maps after day 6 or 7, which is when the animal does pretty well. This is work with Carmena Ganguly back in 2008 when we started this. A couple of controls to show you how robust this retention of the skill is. So remember, this is our circuit in black. Okay, we were talking about stability of the circuit, and this being the spinal cord. So if we really, what we are claiming is that the brain has learned this decoder, right? The parameters that are in that decoder, that kind of prosthetic spinal cord. Well, let's test it out if that's really true. So let's swap in the middle of the experiment, okay, with another decoder, the green one in this case, that the animal has never used for BMI control. However, this decoder predicts arm movement really good, like correlation coefficient 0.95. Okay, so it's what we call a biomimetic decoder, like a linear map of how the brain controls the arm. Okay? But the animal has never used it for this disembodied control. And what you see here is that starting with the normal decoder, the black, blue means successful trials. The animal is really working really hard, performing all trials correct, and then we switch to the other one. It's like flipping the spinal cord to the one that has never been used, and performance drops dramatically. Red means error trial. The animal can barely do it. But very interestingly, this effect is reversible. You plug in, again, the spinal cord that an animal knows how to use, and then performance goes up again. And this is actually very robust. You can repeat this several times, and it's a very profound effect. Another interesting observation is that shows also how, the acquisition of this skill is that once the animal learns one decoder, it can actually learn another one, let's say the green one, without forgetting about this one. If you let enough time, you let the animal learning. In this case, the perturbation was about 20 minutes. And we will swap back to the old one. But basically, if you will leave this one long enough, the animal will learn this new decoder as well. But the point is that will not forget about the previous one. So this is what we tested here. So Carmena, in day 20 after running for 2 weeks with what we call as the old decoder, which is in blue here, that day, all of a sudden, started with a new decoder that predicts arm movement very well, but the animal has never used for BMI control. And you can see that performance is really bad. But all of a sudden, learning kicks in, and the animal is able to get more or less halfway. And then we switch to the old one, the one that the animal knows how to control. And the animal gets a cue showing, like, yeah, now we're back to the decoder I know how to control, right? Like the cursor changes color in that case. And then the recall of the performance is immediate, goes back to 100% and can perform the task. In days 2, 3, and 4, you can see that it's learning how to master the new decoder while not forgetting the other one. And by day 4, it can do both. Okay, so you can switch from one map or one memory to another memory or 2 different decoders for this BMI control. Okay, I'm going to skip this and get to one slide to talk about what happens. How is this learning taking place, right? So now we are getting into can we account in this, exploiting the built-in machinery for prosthetic control? Is this a local learning effect, or is the rest of the brain really actively involved there? So the first thing that we did is to look at, in the other neurons being recorded but not connected to the decoder, what's going on there. Okay, and in one graph, the story, basically, the punchline is that the neurons, which we refer to as indirect neurons, which they're near or far, depending on how far from a BMI neuron are, but basically these 2 groups are the neurons not connected to the BMI. There is a significant differential in modulation depth. In other words, how much these cells fire compared to the ones which are the direct or BMI neurons. In other words, the brain knows which 50 neurons out of the billions of neurons in the brain are the ones that actually are causally linked to behavior. Okay? What's the output of the brain? Otherwise, there will be no difference, which is exactly what you see in early learning. So before learning, you cannot separate with any classifier. There's nothing that really allows you to partition this data in 3 classes. After learning, you have this very pronounced effect that emerges through plasticity after learning to perform this task. So that's first point. The second one is, and I'll try to summarize this a little bit because for the sake of time, this is Erin Koralek's work. It's a neuroscience student in collaboration also with the lab of Rui Costa, formerly at NIH, now at Champalimaud in Lisbon. So we want to test whether deep structures in the brain that are involved in and necessary for natural motor movement, like basically what is referred to as the basal ganglia, several nuclei here deep in the brain, whether these are actually have anything to do during this BMI learning, right? So you have seen all these striking results about this motor memory in BMI mode, but what's the rest of the brain doing, right? Remember, the whole brain is available. We're just using 50 neurons as the output, but we don't know what the cerebellum is doing, what the basal ganglia is doing during this learning process. So here, we target the basal ganglia, in particular one very important structure called the striatum, and we went to the rodent because it's easier to manipulate, and as you will see in a moment, also to even do genetic manipulation with it. So the task here also had a very interesting built-in design in the decoder to force the animal in a way, or bias the animal to not to move while learning to perform the task, right? So what we want is a rat to control an auditory cursor to hit 2 targets, high pitch, food, low pitch, water, or vice versa. Yeah, exactly. Okay, so these animals don't see very well, but they hear really well. So instead of visual feedback, they get auditory feedback, like the tone of a cursor. And then what they have to do is modulate neural activity in the motor area, in motor cortex, in a way that this is the trick that we apply there, which is in a differential way. So there are 2 ensembles now of neurons, and to hit target 1, ensemble 1 has to increase modulation or fire more, and ensemble 2 decrease, and exactly the opposite for target 2, right? And that's exactly what you see there, color-coded for each ensemble, and in red, the red line is the time of hitting the target. Okay. Now, these are cells that control the vibrissa, the whiskers of the rat. So if the rat will try any whisker movement, any muscle twitch as a strategy to drive the cursor, that will cancel the movement of the cursor because all cells will be active. So it has to selectively learn to modulate certain cells in a way and certain cells in another way. Okay? And what I'm showing you here is that not only they learn to do it, and they cannot learn it without visual auditory feedback, but also they're doing it without or irrespective of physical movement. These are accelerometers embedded in the acrylic cap in the head and showing that the animal is immobile before hitting the target, and this is the movement while retrieving the reward, and the same with EMG activity. So point being, to learn to perform this neuroprosthetic skill irrespective of physical movement. Now let's see what happens really in the basal ganglia and these deep structures, because at the same time that Erin was recording in M1 neurons, to control the device, it was recording in the striatum in the same animal, right? And so in early learning, if you look at this coherence, spike coherence between cells in motor cortex and the striatum, there is no coherence, and there is a huge increase in coherence in this band in late learning, which interestingly has been shown exactly the same result in monkeys performing natural motor tasks. So this is what it's telling us, is this a task that is about to control this embodied device without physical movement actually is triggering this plasticity in the striatum, or corticostriatal plasticity in this case, in the same way as it does when it's learning to perform a natural task. Not only that, and this is when the Costa lab became extremely important, they have a mutant, a mouse, that lacks NMDA receptors, which are necessary and very important for plasticity. Right? So the wild type or the control mice that they don't lack these receptors, they get the same result early and late as you see here. Okay? So this is reproducing the same task in a different animal now, in a mouse, whereas the mutants can't learn it. So what this says is basically that plasticity between cortex and striatum is necessary to learn these intentional prosthetic actions, which we think is very interesting from the point of view of the neurophysiology of action. What does it mean, action for the brain? It's not necessarily all about controlling your physical plant, but it's more about getting something done in the real world, with or without your own body. Okay, I will skip that and jump to the last part of the talk. After this is the summary of what we just seen, basically all the plasticity part, both in the monkey and in the rodent. And this second part talks about what do we. So we've been talking so far all about what can the brain do for this neuroprosthetic skill. But remember, we have this decoder here, and we can update the decoder. We can do things with this decoder as well, right? So this field, this framework that Amy Osborne and Sutardja in the lab started pushing very hard about 2 years ago. We call it closed-loop decoder adaptation, or CLDA. It has been going on for about 10 years in different flavors. That people were twitching the parameters of these decoders online in different ways. But what Amy and Sid have been doing very seriously is trying to very rigorously formalize this, okay, in terms of what and when, or when in terms of the timestamp, or when the decoder should be updated, which in terms of which parameters of the decoder, and how, in what form, which rules for update should be the best for, independent of what the brain does, to boost performance, and in very important conditions. For example, when the patient is not moving, is not able to move because it's paralyzed. So if you start from a random decoder or some kind of arbitrary representation, can the animal very rapidly learn that transformation, okay, through online adaptation of these parameters, okay? Online meaning while the animal is engaged in doing the task, in parallel the machine is collecting neural data and what it was going on, what the decoder was doing initially, and using different rules to update them. For example, just to give you two, you could use error signals directly from the brain, which they are in different areas, to decide what was a right trial or an error trial, what the patient really wanted to do or not. Or you could actually exploit the fact that you know what the task goals are. The animal or the patient knows what the task goals are, so you can actually do what we call refits of that activity of the decoder, summarizing this cartoon very nicely. This is the work from, an algorithm published by the Shenoy lab at Stanford. And what it's doing, basically, in black you have the output of the decoder. This is the target that it's trying to reach for, and that's the actual output of the trajectory it's getting. You can see it's deviated from the target. And in red, you have the corrections done by the decoder, meaning you could assume that the monkey, in this case, is trying to hit in that direction. So you can rotate the velocity vector every timestamp and then refit the decoder with that assumption. That's what we call intended kinematics, or cursor movements, okay? So again, Sid and Amy came up with what they call a smooth batch algorithm, which the smooth batch coming specifically for the question on those days we're dealing with when should the decoder be adapted. And you can think, basically, you could adapt all the time or maybe in long batches of 10 minutes, which is how traditionally we have been doing, collecting enough data to do a matrix inversion and being able to predict movement right away there. But they have disadvantages, both cases, which I don't have time to get into, but basically smooth batch, what it does, in this case using this Kalman framework, Kalman filter framework, is to, in very small 1-2 minute data batches, basically update the decoder. Okay, so it's an exponentially weighted moving average of these small averages. In this case, sorry, in this small data epochs, in this case using the refit, but this is not necessary. Okay, we are using this rotation I mentioned before because it works really well, but you could use your favorite way or whichever rule for updating the parameters that you like. But the point is that while the animal is performing the task, okay, in very short time batches, it sees an increase in performance. And remember, these are cases now in which they are starting from zero performance. So when you put them to do BMI, as you saw in that video, they have no idea how to control the cursor. So you want them very rapidly, in a few minutes, to jump up and achieve performance. So for the sake of time, rather than going through the graphs, I'm going to show you the video that basically shows you very well how smooth batch works. Okay? Just want to mention that smooth batch is independent of the initial conditions. This is what I mentioned before. So Amy collected a lot of data starting the decoder in very different conditions, at least 4. One, observing a movie. So it has the monkey just watching the cursor moving and then regressing that to the neural activity and using that as the starting or initial decoder. But you cannot actually do it with random. You can shuffle the parameters and use random decoders and so on. No matter how you start with your initial seed, the performance at the end is the same, so there is no difference, which is very exciting when we think about clinically viable solutions, meaning people that will not be able to move to control the BMI. So the summary of what we have seen before I show you the video: first, adaptation in the brain; secondly, adaptation in the decoder. This is what we refer to as CLDA. Then the ultimate question is how to synergize these two. And now when we get into how to actually design the system to optimize control, there are different signals that you can actually be selecting. LFP, local field potentials, we haven't talked about. We have people in the lab working, like Kelvin and Sid, just showing that you can actually also use local field potentials to achieve the same, not the same, but very good control as with the spikes. You could use different decoding algorithms, and we haven't talked at all. For the sake of time, I will not mention anything about the plant. Okay, the plant is extremely important. Cursors are good for certain applications, obviously, but not for performing tasks of daily living. So you need robotic arms for that. Suraj Gaud in the lab is actually putting a lot of effort in crafting what we call crafting optimal plants, or twitching the parameters, in this case now of the Kalman filter, because we're working with very simple plants in order to basically facilitate what we see in this loop, learning and performance. Okay, let me just finalize with a video in which so a few things are going on here are very important. So this basically lumps the whole talk. Okay, first of all, the neural activity that the Amy is using in this experiment is not single unit activity that we saw before. These perfectly isolated cells. Okay, for engineering purposes or clinical purposes, that's not viable. So now she's thresholding the activity in each channel and lumping together anything above a threshold, which is a much more robust signal in terms of longevity of recording for many years, right? But of course, you wonder whether that will have the same effect as single units in terms of inducing plasticity. Okay, so that's one. And then and the second one is that the decoder here is seeded with visual feedback. In other words, the animal watches a movie for a few minutes of how the cursor moves, and then right after that, the experiment starts. And then, so that's the CLDA part. How to combine this with plasticity, right? So remember, we have these two things. The brain learns, the decoder learns. So what we want to retain from the brain learning really is this natural control, this readily recall motor memory property, right? How to do that if the decoder is adapting, which will become like a moving target, right? So that's kind of like a bit of an issue. So in this case, we thought like, well, one way of doing this will be to, since we are starting from very bad or zero conditions, okay, start with a little bit of adaptation in the loop, in the decoder, enough for the animal to achieve more or less reasonable control, and then freeze that decoder and do the same that we did at the beginning of the talk, right, in the first part, keeping the circuit stable. Same decoder, same signals, long enough to basically for the animal to learn that decoder, and then use adaptation with CLDA when needed. For example, if a channel loses activity or worsens through time or whatever, which will be like a little touch-up to the already existing map. Okay, and hopefully that will allow, the combination of these two factors will allow long-term performance. Okay, so this is what you're going to see now in this story. Sorry, in this video, the whole story I just told you. So the initial performance right after, before any adaptation, is very bad. The animal cannot get to the center to initiate the trial. It didn't hold enough there to trigger it. That's why the peripheral target jumped. This is after one adaptation of a smooth batch. You already can see that the animal has some control. It can hold enough in the center to initiate the trial. It has to hold for at least 400 milliseconds. So it's pretty hard. And now it can reach to any target of the workspace. It's hard, but it can do it. Now, after fixing that decoder for 3 days, you can see that the animal can do almost very similar like the other video that you saw. The animal has learned this decoder, okay, or it's almost done with learning it, and it can achieve targets across the whole workspace. Trajectories are still a little bit wobbly, not that fast, but basically the animal can do it. Now compare with day 13. In terms of the straightness of the trajectories and how goal-directed and the speed of the movement. And remember, this is without the animal moving the arm. This is just looking at the screen, performing the task mentally or in BMI mode. Okay. And again, combining both brain adaptation, decoder adaptation to synergize in this way, so the animal retains the skill of what has been learned, okay, yet also relaxing the constraints about recording from isolating individual neurons. These are now more like noisy channels that we are using. So with this, I don't have time to talk about other exciting things that are going on in the lab. Sorry, maybe another time. just to mention that these are what is left to be done. Again, summarizing now 4 points here of what are the important challenges that some of which I have talked about towards clinically viable BMIs, and obviously acknowledge the folks in the lab again, collaborators, and funding agencies. Thank you very much.